hypersensitivity. Th1 cell stimulates the production of Ig1 and Ig3, opsonizing and complement-fixing antibodies by antigen-activated B cells. These isotypes assist in the kind of cell-mediated immune response that are driven by Th1 cells. Interleukins 2 alone can activate proliferation of Th2 cells and help to generate IgG1 and IgE producing cells. The clonal expansion of activated T cell helps cells is a necessary part of mounting an adequate immune response to any immunologic challenge. The cytokines network that develops will continue to regulate T cell growth and differentiation until the challenge is gone and the response must subside. Transcription of the gene for interleukin 2 and interleukin 2 receptor begins within 1 hour of TCR ligation. The function of interleukin 2 receptor consists of alpha, beta, and gamma or beta and gamma subunits. The beta and gamma subunits increase the affinity of the receptor for interleukin 2 and are responsible for most of the signal transduction through the receptor. The gamma chain is also shared by the receptor for interleukin 4. Interleukin 7, interleukin 9, interleukin 15, and interleukin 21. The importance of the gamma chain is demonstrated in individuals who have mutation in this chain. These persons have X-linked severe combined immunodeficiency syndrome and lack functional T and B cells. Now we're going to talk about the Th2 cytokines, and the Th2 cytokines are the First, we're going to talk about the interleukins 4. As mentioned previously, Th2 cells are primarily responsible for antibody-mediated immunity. Interleukins 4 is one of these key cytokines regulating Th2 immune activities and helps drive antibody responses in a variety of disease. The interleukins 4 receptor is expressed on lymphocytes and on numerous non-hematopoietic cell types. Interleukins 4 activities on naive T cells turns on the genes that generate Th2 cells and turns off the genes that promote Th1 cells, such as interferons gamma and interferons gamma receptor subunits. The chemokines MCP1 enhances interleukins 4 production by naive T cells and could play a role in moving naive T cells towards the Th2 pathway. Th2 cells are responsible for regulating many aspects of the immune response, including those related to allergies, autoimmune disease, and fighting off parasites. Among the many genes induced by interleukins 4 are MHC1, interleukins 5, interleukins 13, and the co-stimulatory molecule B7.1 and B7.2. Interleukins 4 promotes the production of IgG2A and IgE, and along with interleukins 5, drives the differentiation and activation of eosinophil in both allergic immune response and the response to parasitic infection. Interleukins 13 is a cytokines with many of the same properties as interleukins 4, and both cytokines induce worm expulsion and favor IgE class switching. The interleukins 10 has anti-inflammatory and suppressive effects on Th1 cells. It is produced by monocytes, macrophages, CD8 T cells, and the Th2 CD4 T cells. It inhibits antigen presentation by macrophages and dendritic cells and stimulates CD8 T cells. It also induces the production of MHC2 on B cells. However, one of the major effects of interleukins 10 is the inhibition of interferons gamma production via the suppression of interleukins 12 synthesis by accessory cells and the promotion of the Th2 cytokines pattern. Thus, in contrast to most other cytokines, interleukins 10 serve as an antagonist to interferons gamma. It is down-regulator of the immune response. Now we're going to talk about the cytokines associated with T regulatory cells. The third major subclass of CD4 T cells are the T regulatory cells or Treg. Tregs are CD4 CD25 T cells that are selected in the thymus. They play a key role in establishing peripheral tolerance to a variety of self-antigen, allergens, tumor antigens, transplant antigens, and infectious agent. CD4, CD25 Tregs affect the T cell activity primarily through the action of transforming growth factor beta. Transforming growth factor beta induces the expression of FOXP3, a transcription factor that causes T regulatory cells to suppress the activity of other T cells. T regulatory may be found in transplanted tissues and help to establish tolerance to the graft by the mouse immune system through the alteration of antigen presentation. T regulatory are also responsible for inducing interleukins 10 and to transforming growth factor beta expression in adaptive T regulatory 1 cells or TR1 in the peripheral circulation. TR1 cells are CD4 T cells that are induced from antigen-activated naive T cells in the presence of interleukin 10. They exert their suppressive activities on both Th1 and Th2 cells by producing more interleukins 10. Transforming growth factor beta or interleukins 5, T cell suppression occurs through interleukins 10, inhibition of pro-inflammatory cytokines, and inhibition of co-stimulatory molecule expression on antigens presenting cells or APCs, while transforming growth factor beta downregulates the function of APCs and blocks proliferation and cytokine production by CD4 T cells. All of these activities lead to downregulation of the immune response and the prevention of chronic inflammation. These two types of regulatory T cells operate through a network of cytokines to establish peripheral tolerance to certain antigens. Therefore, they play a key role in limiting autoimmunity. The relative importance of each phenotype depends largely on the antigen involved, the context of antigen presentation, and the biology of the tissue. Now we're going to talk about the erythropoietin and colony-stimulating hormone, colony-stimulating factor. The colony-stimulating factors include interleukin 3, erythropoietin, and granulocytes macrophages and granulocytes macrophage colony-stimulating factor or GCF, MCSF, and GM-CSF respectively. In response to inflammatory cytokines such as interleukin 1, the different colony-stimulating factor act on bone marrow cells at different developmental stages and promote specific colony formation for the various cell lineage. Interleukin 3 is a multi-lineage colony-stimulating factor that induces CD4-34 bone marrow stem cell to form T and B cells. In conjunction with interleukin 3, the colony-stimulating factor direct immature bone marrow stem cell to develop into red blood cells, RBC, platelets, and the various type of white blood cells. Interleukin 3 acts on bone marrow stem cells to begin the differentiation cycle, and the activity of interleukin 3 alone drives the stem cell into the lymphocytes differentiation pathway. GM-CSF acts to drive differentiation towards other cell types. If MCSF is activated, the cell becomes macrophages. MCSF also increases the phagocytosis, chemotaxis, and addition cytokines production in monocytes and macrophages. If GCSF is activated, the cell becomes neutrophil. GCSF enhances the function of mature neutrophil and affects the survival, proliferation, and differentiation of all cell types in the neutrophil lineage. It decreases interferon gamma production and increases interleukins-4 production in T cells. And it mobilizes multi-potential stem cells from the bone marrow. These stem cells are utilized to repair damaged tissues and create new vasculature in these areas. These activities are necessary to reconstruct tissue following an infection. However, interleukins-3 in conjunction with the GM-CSF drives the development of basophil and mast cells, while the addition of interleukins-5 to interleukins-3 in and GM-CSF drives the cell to develop into eosinophil. The net effects is an increase in white blood cell to respond to the ongoing inflammatory process. EPO regulates RBC production in the bone marrow but is primarily produced in the kidneys. The protein in its physiological form is a 34-kD monomer with a high carbohydrates content. Glycosylation of the protein is required for activity and accounts for the structural difference between recombinant EPO alpha and EPO beta. Both proteins have the same amino acid sequence and similar activities as endogenous EPO, but EPO alpha is the form licensed for clinical use by the FDA. EPO alpha is often prescribed to improve the red cell counts for individuals with anemia and for those with cancer who have undergone radiation and chemotherapy. RBC proliferation induced by EPO improves oxygenation of the tissues and eventually switch off EPO production. The normal serum EPO values range from 5 to 28 UL, but must be interpreted in relation to the hematocrit, as level can increase by up to 1,000-fold during anemia. Cytokines. Now we're going to talk about the cytokines and anti-cytokines therapy. So cytokines inhibiting biological disrupts the interaction between cytokines and their cognate receptors. Initial studies use murine monoclonal antibodies that were able to function only for a short period of time before the host mounted an immune response against them. Recombinant DNA techniques have allowed for the production of humanized monoclonal antibodies that are much less immunogenic and function as cytokines antagonists. An example is infliximab or Remicade, a chimeric antibody containing human constant region and murine antigen-specific arms that bind human tumor necrosis factor alpha. Remicade blocks the activity of the tumor necrosis factor alpha in rheumatoid arthritis and Crohn's disease. A single dose alleviates symptoms and reduces swollen joints in RA patient for up to 4 weeks. Multiple infusion achieve in 60 to 70% improvement in symptoms in this individual. Monoclonal antibodies can be further humanized so that the complementarily determining region are the only murine components remaining. Zenapax, which is direct at human interleukins 2 Rx, this antibody generally lowered affinity and extended half-life, but reduced immunogenicity. As an alternative, transgenic mice that have human immunoglobulin genes are able to produce fully human monoclonal antibodies from mouse hybridoma cells following immunization. These antibodies have high affinities and long half-lives and are not immunogenic.